We've reserved the rest of, or much of the rest of the afternoon for some roundtable discussion. Mel Vargas is gonna lead that and help hopefully bring all this together, and maybe we can discuss, how this applies to our watersheds and maybe what other questions this has brought up during the day. So, Mel?
Okay, well, had a good round of presentations there. I really appreciate the effort everyone's put into them. Yeah, you can turn that off if you want or whatever. So I guess to start this next 25 minutes, first I'll just ask if there's any question that someone's just got to get out right this second that they didn't get to ask earlier. They really want to follow up on something that didn't get fleshed out maybe enough earlier. Can start with Jill.
This isn't a question, but it's a comment based on something that was asked during the first talk, and that was, what's the sampling guidance regarding bridges? And the Swiftham's Procedures Manual does specify that people should be collecting their samples upstream of a bridge's influence, not downstream. It's in Chapter 3 and 5.
You also mentioned inflowing water, right? Yes,
inflowing water. And sometimes people have to take samples from a bridge, but we ask them to take it on the upstream side of the bridge.
So there's a decent cross spectrum here today of river authorities. That's what the river authorities are doing, correct? See a couple nods. Where's the LCRA folks? Yep. I saw the Sarah folks. BRA, okay. Who am I forgetting? Debbie? Yeah. Okay. Okay. Other questions that maybe didn't get explored enough earlier on any particular topic? Way in the back.
I'm curious after listening to the presentation earlier about the way that the high resolution sampling changes some of the correlations that you see between other parameters. And I'm kind of curious on you guys' thoughts about how that might affect our models that we use for explaining soil water quality management. Because, as you know, a lot of models are based on these very low resolution sampling.
Yeah, there's lots of folks in here that have had to utilize different models to get them where they are currently in their watershed protection planning process. And I can assure you that each of the folks, if anyone wanted to take a stab at, sharing their modeling experience on their watershed, yeah, they're all dealing with, I think, a common concern, which is, I'm just going to use the word limited data. It's limited in that most data that's gone into models that I'm familiar with on most of the watershed protection plans around the state to this point are monthly at best, quarterly usually. Sure, lots of watershed protection plans may have a focused monitoring effort, but still that monitoring effort's probably at best, what, if you're lucky, 2 years of data at the most, probably, that got funded as part of the watershed protection planning process. And even that's probably still going to be at best monthly, unless you have some storm events thrown in there when there is one that, you might get 2 events in a month. So I would say it's generally, I think everyone would agree that watershed protection plans to date across the state have certainly not been able to benefit from high-resolution monitoring plans, if you want to call it that, as you saw in this presentation earlier today by Hanadi. Some models are equipped to do daily time steps. Some of them aren't. Yeah, I think it'd be great if we had more data to go into these things. Unfortunately, money's a problem usually when it comes to the monitoring plans. Is there anybody else out there that I'm missing that's done modeling for watershed plans that has more data than just monthly or quarterly? You know, a tighter, higher temporal resolution than what you know, something on the order of what you saw up here? Nobody's raising their hand. Kirk, which one specifically?
When we have a model, we never specifically go out and calibrate the model. We do try and address at least a few runoff events where we have a few data points over the course of the runoff event. And at least with the HSPF model, we can calibrate to some data.
Yeah. Right. Okay. Bill?
In doing that sampling there, I think that can give you some insight into that variability in your monthly samples of seeing where they occur in the day and bring a little context to saying, well, okay, I've got a few that are around this way, and see if you've got that afternoon, evening, early morning difference that's causing that. There's also a temperature swing through the day that will cause some numbers changes. So I think that looking and seeing if your watershed has a diurnal impact, it could reflect on it, whether it actually went into the model or not, but it just explains some of your adjustments to your model.
Yeah, that's a very good point. I mean, looking forward, that's what the purpose of these presentations are, sharing technology, information. It's the first time I've seen something with that pronounced of a, I guess, bacterial diurnal response, and that, to me, tosses it out to all of you experts. Maybe the next time you sit down and do your next monitoring strategy for a watershed project, I don't know, maybe you should consider applying some of that money to a higher resolution focused effort to see if it can or should, like Phil just suggested, influence your 12-month or 24-month sampling strategy that you're going to do, because you might adjust when you go out and collect those weekly or monthly samples, whatever you're going to do. First time I've ever heard that. It's probably not a bad idea. Consider it, deliberate it. Rocky? You want to adjust.
Switch around
Yeah.
the order of sampling,
Yeah.
but it's usually because it's most efficient because we have to get back and get samples to fly at the time. So.
I've scribbled down some thoughts here that I'm just going to toss out intermittently here, but since one of them actually has to do with Rocky's presentation earlier, so now that I've got you, your attention and you're on the spot here, I guess I'm curious, you know, your thoughts about, in your discussions with TCEQ, about potentially rethinking the indicator that you're using to evaluate whether the watershed protection plan implementation strategies are effective and are going in the right direction. I mean, this idea of shifting indicators to maybe in Terracosi for the Arroyo, I'm just curious what that means to the 10-year time frame of the way the watershed protection plan was initially developed and written up. Here you got this document that, you know, everybody's kind of signed off on and says, okay, go forth, do good, and make sure it works. And you better come back with results. And it's based on an E. coli target, and now you're suggesting maybe the target might be rethought. You know, it brings up this whole idea of adaptive management, and we're all struggling with what that means and how to apply it, and I think we're really struggling with it from a institutional perspective, whether it be the federal agencies that have to respond to it or the state agencies. I just wondered if you've kind of started to play out that painful discussion.
Well, personally, now, I'm not as involved in the watershed planning, but the reason it got started, I mean, for the Arroyo, the reason it got started is, okay, here we had, you know, the E. coli gets assessed in 2 assessment units. It meets the GME, but yet we're not going to use that data. So that's so why are we collecting it if it's not going to be used? And that's what just started the whole initial process of thinking. But is Alan, he's working with more closely with the Royal Watershed Partnership, and he may be able to answer that question about the adaptive managers that are 90 Sorry, Alan.
Yeah. It's not, yeah, I know there's not a specific answer, but I think I want everybody here to be thinking, I want to hear from you, Alan, but everybody needs to be thinking about these complexities that, this is the first time I've ever kind of heard this idea of maybe potentially the target or indicator changing after you've spent a lot of focus and energy on one. So, go ahead.
This is honestly the first that I've heard of it. Working with Jaime, I mean, he's got more background on the project as a whole than I do. But the initial targets have been nutrients. We haven't focused a whole lot on bacteria. We've all done more with the RUAA. There's been, I think it's 12 out of the 14 wastewater treatment plants updated. And so, I mean, but that's about the extent of focus on bacteria.
Okay. Okay.
Lately,
Now, the EPA is looking right now at the question of indicators. Are they supposed to come out with the results of their studies either this fall or next fall?
Well, last fall it was the fall before, and this fall it's next fall, so I'm not sure whose fall it is, but I guess I was actually kind of hoping we could at least just get a little update on that from someone here from EPA. And I mean, I don't want to go far afield on this tangent of water quality standards revisions and criteria changes, but if someone from EPA is willing to just comment on where that whole WRF EPA project is, it really more from the perspective of when those types of reports are available for the people like in this room and the practitioners to get their hands on. And again, I don't want to go off into the water quality standards tangent. I just know there's a lot of really valuable information that they're looking at in terms of sampling methods and questions related to the indicators. And if anybody from EPA was able or willing to just give a quick couple statements on where the WRF EPA effort is right now. Okay.
Good afternoon, everybody. I'm Jane Watson, Regional Office.
I didn't even see you, Jane. Jeez. Sorry. I apologize.
We are slated at this point to release the new recommendation and complete with the studies October 15th of 2012. And now there is there was a lawsuit with surprisers, and there was a settlement agreement, so that's part of a commitment to the court. So it's not, a totally flexible thing. So there will be, the report to the court, and then that will be made public. EPA's been doing some stakeholder listening session updates. They did one in Chicago, they did one in New Orleans, in which they gave updates on where the studies were going and what the current thinking was coming out of those. And when I get back to the office, I'll see about getting the website, because those PowerPoints are up there on the website for folks who weren't able to, like me, who weren't able to attend those. But it gives some discussions and some progress. They did a lot of work. They refreshed the epidemiological studies at Great Lakes beaches, some on the Atlantic coast, two on the Gulf Coast. They looked at a tropical and a subtropical beach, that being South Carolina, Puerto Rico. They looked at a beach that was impacted mostly by urban runoff, because that was one of the questions that we agreed with the litigants that we addressed. They looked at a spectra of different organisms, very similar to what they did in 1986, but they looked at that. And they also, what part of the settlement report is that we would look at some very rapid method as well.
Okay.
That would be more useful in telling somebody if a beach on a given day was getting bacterial intrusion or whether it wasn't. What came out of that, there's a method called polymerase chain reaction that will burst the cell and extract the DNA from enterococci and get a signal. So it's a 2-hour test, and that they got pretty good behavior with that, linking up to the illness rates that they did. So that's something that's probably going to be part of the recommendation and be something for a short-term type turnaround. Some of the things that will probably look different than what's currently on the books is the single sample maximum idea will be replaced with something called a statistical threshold value. There's still going to be a recommendation for a central tendency or mean bacteria level, and then this statistical threshold value, but probably going to get away from this use intensity concept that you can select a number based on if you thought the beach was heavily used, moderately used, lightly used. That concept will probably go away. But basically, a lot of what these studies show is that the only 6 numbers in that concept were reasonably robust. They're not seeing that cultural E. coli they're seeing is still a pretty strong indicator in the form. Now they changed up, what the trigger for illness. So it's a larger group of people that they call getting ill because you didn't have to have a fever to go into the ill bin. So it's a broader definition of illness, and that might be a communication challenge.
Yeah.
Because all of this is always a communication
challenge. Yeah.
Most people intuitively don't think probabilistically.
Right.
Yeah. Or would they do anything else? But it's it's anchored. It's going to be anchored in the same illness rate, so it's the same level of protection. It's a very important thing to the court. The court had asked us to look at if we could do something that was children-specific. In the data, there's not enough data or robust enough compartment of just children to do that. So the studies using the general population include children, so that we're looking at that and being protective of everyone. We also did not get information that would allow us at this point to make some general nationwide determination of the difference in risk from human pathogens versus animal pathogens. The information does not lend itself to that. And that was something that all these studies attempted, but there's not the appropriate information to draw that conclusion.
Okay.
They did something called a quantitative microbial risk assessment, which is QMRA, to try to get a handle and learn more about the relationships between bacteria from any or either source and illness rate. And that may be, there may be some more information come out on that. That may be a technique, albeit expensive and difficult, where states might look at some site-specific difference in illness rate. There's an example there that's going to come out. But basically what they did is they went to a site that was downstream of a CAFO. They measured both the indicators and the specific pathogens, like norovirus, rotavirus, adenovirus, crypto, giardia, Campylobacter, Salmonella, and E. coli, things that would come both from humans and animals, and then have a better understanding preponderance. So, for instance, there's a significant animal source of Cryptoma giardia, as an example. And they looked at the different predicted risk of illness levels from these animal pathogens. The ones from cattle come in overlap. The upper confidence bound overlaps the risk you get from POTW effluent. Okay, so that's for that kind of dispels the notion that cattle waste is a lot less risky to you than human waste. Didn't come out that way. Chicken waste came out from that type of modeling with a little lower risk, but the cattle overlapped and the pig brushed right up against the human exposure risk. So you will, there was an attempt, there will be some much more sophisticated thinking that will come out of these recommendations. And being that nobody wants to be contempt of court, it will be out next fall.
Okay. Thank you very much for that. Appreciate it.
I want to comment. At the Chicago meeting, I think there was one person from Texas who attended the stakeholder meeting, and I haven't asked about the New Orleans meeting, but when I called, there wasn't anybody who's registered to go when I called before. But that's great. So they told me there was only one person.
Okay. I'd like to kind of pose another question here. Listening to the presentations, particularly, I don't know, just Dr. Gentry's, I guess, presentation and Kevin Wagner's presentation, because they were in some of their sites were in a watershed that there was a watershed protection plan done, the Leon River watershed. I was churning through my brain the whole time trying to figure out how do we take this information now and convey it to the stakeholders who did not have access to this information during the watershed protection plan development process, and what does this data from each of these three disparate yet related studies really mean? And I may even have forgotten one. I mean, Emily's work with the different sampling techniques. And I'm not sure I know how to do that. I mean, I'm not sure yet what the answer is to that question. How do we go back and what did they learn from this? And I want to kind of put somebody on the spot here only because they might be able to help frame my question better, and you can certainly decline if you want to, but Commissioner Clary, I was just wondering if you might share with us how you think the ranchers or the farmers or the county officials might react to just some of this, just at a high-level reactionary perspective on what some of this data you've learned, and if you care to share any of that with us, or for that matter, you, Trey, as a representative from Brazos River Authority. We've got to find a way to get this science better used and better understood.
My quick comment would be, they won't understand it. I'm sitting here as an uneducated individual interested in the subject, and even though I'm struggling to stay up with the group. So I think the confidence that the local stakeholders have with those who they are working with, will be very valuable. Where that water quality professionals that are helping them solve their problems will be able to present this information in such a way that can help them understand, help them work through it. But they're not going to understand most of it.
Yeah. Trey, do you want?
I think it might have to do with one, Mr. Howard, if we hire a watershed coordinator, make sure they understand that information.
Absolutely.
And also, the buzzword that Jay Bragg talked about, the adaptive management portion of that is, as time goes on, your information is better. You see what works, you see what doesn't work, what does not work. If there's new techniques, you integrate that plan, they can adapt your plan to facilitate that.
One of the things that I heard earlier today And, you know, the watershed protection plan is just simply looking at a goal, water quality goal, of reducing bacteria. You really don't have any idea which best management practices have been effective in those ones on.
Right.
Whether it's in agriculture, you know, been effective with the urban, being been effective, or whether you have some work with non-point sources. You won't know without finding out a way to distinguish between sources and their contributions.
Yeah. Yeah, I mean, Dr. Barrett's presentation to start out the day on the swallows under the bridges, you know, even that kind of information, I wish I would have had that type of information at my fingertips earlier, but it wasn't available then. So I guess I just, you know, all we had at the time was we had a quantification of the number of road crossings in the watershed, but we had no way of knowing how many birds that equated to, or even remotely how much you know, fecal coliform production was generated by bird populations at bridges. But having a study like that, even though it's just in the Austin area, I think that would have helped put things at least in context if I would have had that information at the time when we were working on the Leon. And I just, that's what I challenge people, I guess, in the room to do, is figure out a way to bring some of these results back to their watershed and convey them in a proper manner as good examples. I guess looking at the clock here for us to, wrap things up here and get out of here by 3:30, I guess I'll just kind of close it up by asking if there's any other, just last question that someone's just dying to get out there and get another little discussion on. Okay, well, I'll turn this over to Kevin, and you can kind of talk through our next steps. And, you know, I guess I'm going to give the last question, and it literally is a question because there's not an answer to it, but it might be a topic that needs more deliberation in the future. One theme that I heard in every single presentation today, there seems to be a very strong relationship between bacteria concentrations, loads, I don't really care how you want to express it, and sediment. Whether, and you can define the sediment any way you want, either bed load sediment or watershed loading sediment. But every presentation had some interconnection between sediment and bacteria, and you know, it came up again at the last watershed stakeholders meeting we had when some of the individual presentations were made on watershed protection plans. I remember very distinctly people asking after 2 or 3 of the presentations were done, they just asked the question, well, did you look at sediment loading? And the answer was no every time. No, because of a lot of reasons. Sampling, lack of data, budget, complexity, and maybe the most important one, thank goodness, I'm saying this and this is probably good, there's no regulatory aspect to dealing with sediment management. And I'm saying that from my perspective. I know there's a lot of different people in this room that maybe have a very different opinion, that there does need to be sediment management tools, as in some type of sediment criteria for streams or lakes, but it's a nebulous, complicated, long-term issue that we're going to have to deal with, and it comes up every time. And I didn't say I had an answer, it's just an observation that it's there, and I think we need to look at how we're going to deal with that on a watershed-by-watershed basis as we undertake these watershed management planning processes. So, that's it for me.
All right, thanks. Go ahead, Jay.
I want to say one thing, or a few things, I guess. When we're out going to the top, working these watershed protection plans, and asking producers to implement management practices that are going to cost them dollars to be a producer or be a city or whatever. We need to have some kind of assurance that whatever we install is going to benefit water quality. with the presentations we heard today, there's still so many questions. Not saying that they shouldn't do anything, or that they should do anything, but we need to have, there needs to be some kind of accountability, we need some kind of assurance that, yes, if I spend this much money, I'll get this kind of production. And I don't think that we can say that right now. Okay.
Well, there's certainly a lot of variability, no doubt about that.